Good morning, everyone. I'm Bob Blankenship, and I'm happy today to be able to introduce Hao Zhang, who will give our PARC seminar today. Hao is a native of China and got his bachelor's and master's degrees in China at the East China University of Science and Technology and the Hebei University of Science and Technology. He then came to the U.S. and got a master's degree at the University of Wyoming. And then came here to WashU and got his PhD under the advisorship of Mike Gross in the area of mass spectrometry. That's when I first met Hao. He and my former student, Jianzhang Wen, were good friends and worked together to really bring our research groups together, and has sort of served as the foundation of a fantastic collaboration between our group and the Gross group that has been phenomenally successful over the last several years. And one of the most exciting aspects of PARC has been the fantastic contribution or impact that mass spectrometry techniques have made on research in this area. And a lot of this work has been really due to Hao's imagination and very hard work and diligence. And so he's now the director of our mass spec facility within PARC. We have one instrument that is part of the PARC instrument collection, and we have a second mass spectrometer that is requested as part of our renewal application, and we're optimistic about that. And we've sort of become now integrated into the mass spec resource facility in the cyclotron building that Mike Gross is the director of. And so it's been a very, very productive collaboration, and Hao is going to tell us about some of the work that he has been involved in, and some of the future work that is going to be done here in PARC. Hao?
Thank you for the introduction. And and I also want to thank PARC and Dr. Gross for providing me this opportunity to have a chance to discuss all our related work by using mass spec and study the protein systems in the photosynthesis with the audience and all the PARC members online. The PARC, we are interested in the photosynthesis. We want to use the different technology to understand the mechanism involved in the photosynthesis. The one thing we focus is called the antenna system. You can see here a couple of different examples, the different photosynthesis systems in the different organisms. The one thing in common in those systems, they have one part of the protein complex called the antenna. The basic process is collect light and transfer the excited energy to the reaction center. So how we study this antenna system? The one part of the modern biophysics is studies relating the structure or dynamics of micromolecules like protein complex to their function. Like I show here, in that study, we focus on the high order structure of proteins, like our favorite protein, the FMO antenna system. We really want to understand the protein structure and the related functions involved in the photosynthesis. How we can study those things through the biophysics, especially the mass spec biophysics approaches. They indeed have a lot of traditional tools in the biophysics to study the protein high order structures, like the CD, IR, light scattering, SEC, and UV, and so forth. And they can cover the different level of the structure of the protein complex or protein. If we're talking about what kind of information we can get, we can see those methods very easy to use. But the information is limited. They only can provide a certain level of information. Of course, in the research field, we do have a high resolution method, either X-ray crystallography or NMR. They can provide a high resolution structure model, but the limitation is high. A significant amount of effort needs to take to reach the high resolution structure models. And also in many cases, the protein system cannot be crystallized to meet the requirement for the X-ray. And also, so many systems cannot reach that purity and the size to meet the requirement from the NMR. Very clear, here is a gap between the high resolution method and the traditional low resolution method. The mass spec-based biophysics tools fit in this gap very well. In the mass spectrometry-based method, we can study the protein in their native state in the solution by using the footprinting technique or the native mass spec, and also the newly development, the top-down, where the native mass spec, to give you the information from the global level of protein conformation down to the residual level information. These mass spec tools fit very well between the traditional low resolution tools and high resolution tools, and also give you the complementary information you can use to combine all the information together to generate a useful understanding about the target system. Here is our favorite protein, the FMO antenna complexes. We have been studied for a long time. And in this talk, I will be using this FMO antenna complex as an example to go through with you all the methods we used, we developed, and some methods still under development, but already generate useful information. In the biophysical approach, mass spec-based methods, we can divide it into two types. One is the bottom-up type, which majorly involved denature the protein and using different enzymes to cut the protein, like trypsin or pepsin. By that way, we are looking at the peptides. All the information measured by the mass spec is peptides, and all the information generated based on this measurement. So how we can do this? Here's a major tools we use called protein footprinting in this technique. And here's the example. For example, we have the protein complex and the protein single subunit here. And both of them can go through the labeling, the footprinting tag. And either chemically or using some enzyme, we can label the surface or the solvent accessible area of the protein. You can see right away. Between the different samples, we can generate a different labeling result. For example, the interface within the protein complex were protected from the labeling, and those parts is free from labeling. Where in the single protein subunit, those parts can be labeled. And then we do the denature of the protein and the digestion. So we end up with the peptide mixture, and within the traditional bottom-up proteomics, we can analyze each peptide and read out the mass. The simple mass shift between the unlabeled peptide and the labeled peptide can tell the information where is the interface. That's the basic of the protein footprinting method. But the question is, how do we label those proteins? The easiest way has been developed for a long time. It's called a hydrogen deuterium exchange. They can target on all those exchangeable hydrogens among the peptide chain. And based on the readout, the different mass between the deuterium and the hydrogen, we can read out the mass shift and generate the information about which part is protected, which part is not. One of my former colleague, Richard, first using this method to study the interaction between the base plate proteins and FMO protein. And by comparing the exchange result between the isolated FMO protein and the FMO protein with the peptide from the base plate, he can locate the interface between these two subunits. And this paper published in the biochemistry. But after you see that system is sit on the membrane and have a multi-subunit to form the whole photosynthesis system. It's complicated. So one way we want to look at that way is we're using very simple modification method, and the result can be really easy to explain. That's the way we developed in our lab. It's called a target to the carboxyl group. And because we're using glycine isoester to label the aspartic acid or glutamic acid in the ammonia acid side chain, so we call this method in short called GE labeling. The labeling using the two simple reagents, the EDC initiate the reaction and the final attachment of the glycine isoester to generate the formal product. And the mass spec can readily to read out the mass shift from here and give us the information where can be modified, where cannot be modified. We started working on this system. As I mentioned, you can see here, this system is quite complicated. And we have a big, giant chromosome on the top of this FMO protein, and the membrane embedded the reaction center inside of the membrane. The first thing we want to understand is how this FMO protein interacts with the base plate protein, like the information provided by the HE chain from my former colleague, and also how to interact with the reaction center. Why such information is important? If we look at those FMO proteins, it's a trimeric complex, and within inside those white molecules is a pigment. Those part is a major player for the energy transfer involved in the FMO protein. And people believe the energy transfer is involved the different sequence of those pigments. The one thing we can understand of this orientation, we can generate the information about how well those pigments involved in the energy transfer, which one have a higher energy, which one have the lower energy. As Bob mentioned, my colleague, Dr. Jianzhong Wen, actually prepared those samples to study this labeling technique. We have the sample, either the isolated FMO protein or the FMO protein from the chromosome depleted membranes. That means there's no chromosome. All the FMO from the native membrane part. If we label them with the GE labeling, the expected result should be the isolated FMO all over the surface available for the labeling. Well, the membrane attached part should be protected in this sample, and the both parts should be protected in the native membrane system. The result end up like what we expect. You can see the labeling extent among the couple peptides, which is on the bottom part of this FMO protein. And you can see the only higher percentage of modification happens isolated FMO. But for the chromosome depleted membrane and the native membrane, the modification level is low. That indicates those peptides actually sitting on the interface between the FMO and the membrane side, instead of the side between the FMO and the base plate. We generate this first experiment evidence about the membrane orientation of this FMO protein on the green sulfur bacteria photosynthesis system. And this method is quite well to fit our studies about all the biophysics with the different membrane system or soluble proteins. We further continue those works with different collaborators and with interesting proteins like involving the disease and the method another one, and also the method development. And also our PARC member also started using this method to study some information from other systems, as well as some system involving the disease. That's method is work out quite very well, and we are willing to continue to using this method and extend the method. If we're looking at detail of this FMO complex, again, this time we show in the green color is all the pigments. But in the bottom-up approach, we denature the protein, and we analyze in peptides of those proteins. All the information between the pigment inside each of the monomer of the FMO is get lost during the process and purification because we destroy all the non-covalent interactions. Is there any way we can look at them intact and generate information about the global level of this protein complex? Because this one is the function unit to function in the photosynthesis system. This come out with the other approach we use in the mass spec. It's called top-down, where the native mass spec. Through the native mass spec, like the name, we don't denature the protein, and we don't use the enzyme. What we can get is the intact information about this function unit and the protein complexes. How we do that? We can introduce the whole complex into the gas phase by the native mass spec, which we're going to cover shortly after. But if we can introduce this one into the gas phase intact, with all the non-covalent interactions still intact, we can read out the mass of the whole complex system, and directly we got the information about the complex stoichiometry. We have another technique available in our PARC machine, mass spectrometer, is the ion mobility. This ion mobility technique is similar to the native gel we used all the time. That means the protein complex ions go through the field with the electron field to push, go through from one side to the other side. In the meantime, they face a lot of clearance from the gas molecule inside of the drift tube. The larger or big size of the ions will suffer more clearance and travel slowly, and the smaller and more compact ions going to travel fast because they experience light clearance. Based on the arriving time on the detector on the other side, we can generate the information about the size or the shape of those complex ions. We also developed some technique by using the dissociation method available in the mass spectrometer, like electron capture dissociation, to fragment the flexible region on the complex without breaking the whole complex. Those fragments can be read out, identified by the mass spectrometer, and generate the information about where is flexible regions. I will use, again, the FMO as an example to go through all those applications. So first, how we introduce to the gas phase the intact complex. Here is the regular ESI spectrum of FMO protein. We all know that if we denature the protein, the only thing left is the unstructured polypeptide chains from the one single subunit. The molecular weight is around 40 kilodalton, and they can carry a lot of charge because they denature. All the positive charge residue is available to carry the proton in the mass spec. We end up only read out the mass of a single subunit of the complex. But if we're using non-denatured situation, mainly through the water-based solvent with a certain salt to contain the ionic strength in the solution, to introduce this one to the mass spec, we can introduce the whole intact protein complex into the mass spec. The readout looks like the protein carry less charge and fall apart in the high MOA range, but the molecular weight this time is 141 kilodalton. It's consistent to the whole trimeric structure, within all the pigment still inside. This gives you the opportunity to transfer this complex into the mass spectrometer and open for all the methods you can use to interrogate this complex. So we do the example, we analyze the sample from the FMO protein With the higher concentration, we do see the two trimer FMO protein from the dimer in the very small amount, but majority result is the intact trimer of FMO. It represents the intact protein complex. If we apply the energy, we can initiate the dissociation of this protein complex and get the unfolded monomer signal here, as well as the pigment bacterial chlorophyll A inside. The first impact of this result is the question about that mystery ACE pigment on the top of the FMO system. There have been a long time studies focus on the whether for this FMO system, except 7 pigments inside of each subunit, there's a one extra one on the top of the FMO complex for each subunit to involve in the energy transfer. Unfortunately, the available high-resolution X-ray crystallography cannot identify or confirm the existence of this ACE pigment. In the mass spec, when we introduce this mass spec, the intact complex into the gas phase, the first thing we can do is to measure the mass. And based on the mass, especially for each charge state, you can see the multiple peaks observed in each charge state. And the mass difference is quite consistent. It's related to the head group of the bacterial chlorophyll A. And the overall mass is very close to what we expect if we have 3 more pigments for the whole system and involved in the intact FMO complex. This provides the first experimental evidence the existence of the X pigment and help us to understand the energy transfer actually involved in this FMO protein complex. We also can do, as I mentioned, is the M mobility. In the M mobility, we can using the arriving time or drift time information to separate the different shape of the molecules. So here, the example from the FMO sample, we already know we apply the energy. We introduce the unfolding monomers. You can see not only by mass, but also by the size, we can isolate those intact trimer and also unfold monomer. Because the unfold monomer have a different conformations in the gas phase, we do see the large distribution in the arriving time compared to the intact trimer. Thus, give us another opportunity to directly study those protein conformations or protein structures by using this new technique we have in the PARC. So we also want to develop a new technique to understand more on this context in terms of the fragmentation. We shift gear to the high-end, high-resolution instrument called the 12T FTICR mass spectrometer from the mass spec facility directed by Doug Gross here. And this instrument from the broker and supported by the NIH funding. But in this instrument, we have a multiple method working on the dissociation. For example, the clearing cell, you can do the CID, or in the FDICR, you can do the ECD or IRMPD. The typical for the electron transfer dissociation, you can introduce very fast fragmentation by electron attach and form the typical Z and the C ends among the peptide sequence. We first developed this method using the model protein called the alcohol dehydrogenase tetrameric protein, and we call ADH for short. And we introduced through the native mass spec, we see the similar thing as the FMO protein is a tetrameric form and which with very less charge state. After we applied the ECD, what we observed, not only the charge reduced form, because we attach the electron to the positive ions, we neutral some of those ions and get the charge reduced ions here. More important, we see a lot of fragmentation in the lower M of the range. With the high resolving power from the FTMS, we'll be able to identify all those ions. It turns out all those C ions come from the N terminal of this protein. If we label those N-terminal sequence ions on the structure of the ADH, all those parts is on the surface of this tetrameric complex. This gives us another opportunity to generate some structural information about where those regions on the surface of this complex can be directly read out by the ECD native mass spec top-down experiment. So why those parts be fragmented? It's not only they are on the surface, also they have a relative higher flexibility. For example, here, this is the plot of the B factor used to measure the flexibility through the X-ray crystal structure. And you can see the region we observe in the ECD top-down fragmentation is actually have a relative higher flexibility among the whole structure. This again confirms our assumption. This ECD top-down method can generate the structural information about where is the flexible regions about this complex. We then apply this to our favorite protein FMO complex. What we got is this time now the Cn from the N terminal is the Zn from the C terminal. And also with the combination different dissociation method like CID, we got another coverage also from the C terminal part in the middle of the C terminal domain. And based on those information, we can generate is either this part or this part is involved in the highly flexible region in the FMO complex. We go back to see the B factor, the flexibility of this whole complex. We can see the region we cover actually is one of the highest flexible regions among the whole structure. This result confirms our previous explanation for the ADH and further gave us the opportunity to using this method for more and coming protein complex systems working on and generate useful information using this new technique. So overall, we can summarize here. Working on the FMO complex protein, we either can go the bottom-up approach. We're using the protein footprinting and denature the protein using the enzyme cut to the peptides without the modification. Generate the information between the protein-protein interaction and the orientation of the membrane on the membrane of this FMO complex. In the other way, we can transfer this protein complex into the gas phase intact and generate information about the intact function unit and the protein assemblies, more important the structure, like where's the flexible region of this protein complex. If you think about all those methods we use, it's kind of a toolbox of the mass spec-based biophysics toolbox. That means we can use a different approach, either the bottom-up approach to study those interactions, and we also have some development with the cross-linking part, and also the top-down approach. And we're mainly talking about where the native ESI and to give the direct measurement, mobility, and ECD top-down. This whole toolbox, most of them work very well with the FMO protein. More important, those things can be extended to any system we are interested in, for example, in the photosynthetic system, to generate comprehensive information, what we can get from the mass spec. They may be not very high resolution, but they can fit very well the gap between the X-ray or MR structure if they are available. If they are not available, they can generate also very useful information here, as well with the low resonance method with the different directions. So we have all those methods in-house, ready to go. And the one system we further studied is called orange carotenoid protein, OCP. This protein has been reported their structures and the function from the different groups previously. The major function of this OCP protein is involved in the quenching of this cyanobacteria photosynthesis. The study shows when the system receives a strong light condition, the OCP will bind to this system to initiate the quenching, to release the energy as the heat to protect the reaction center intact in that saturated light condition. And in the dark condition or less saturated situation, this protein can be released by the other protein and to restore the whole activity of this photosynthetic system. How this protein evolve in this way? The previous study shows the protein has two conformation or two states. One is they call OCP-O, is orange form, and the other called OCP-red, is red form. And here is the different absorbance spectrum of these two dark form and the light form. The inactive OCP-O form, when they receive the light, they can transfer or convert to the active form and called OCP-R. But there are little known about what conformational change involved in this transition and what actually happened on this system. We're using the mass spec to give a look. The first thing, the simple way we can look at this system is the native mass spec. We introduce this system to the mass spec, directly measure the mass. In the dark adapted form, this orange sample shows actually the monomer and dimer mixture. When we illuminate the sample with the strong light, the sample changes to the red color. And the mass spec shows the majority part becomes the monomer. And also the monomer has the chance to carry more charge. That means this monomer is quite open, and the majority part is the monomer. If we put this sample back to the dark situation, we can see gradually the dimer peak come back. I mean, in the mass spec, we can monitor this transition from the dark form to the light form and all the way back to the dark form. This first provides evidence there actually is a monomer dimer conversion in this function. And also, we take advantage of the M mobility to measure the fat change and using the clean energy to initiate the dissociation of those monomer and the dimer. Here's the drift time, arriving time from the drift tube. And you can see the dimer peak shows gradually open and into the other state and lose the pigment, as well as for the dimer. But the one important thing is here, the monomer actually start open earlier compared to the dimer. This maybe suggest that the monomer is more quite open compared to the dimer and willing to share the pigment to involve the quenching of this protein's function. Those information can be very quick to generate by the mass spec and use very less samples. The other question we want to answer is where is the dimer interface? We pick back our protein footprinting technique called GE. Again, here we're looking at the dark and the light form. Here is the result from the different peptides of the modification percentage for different situations. The dark form is in the blue and the light form is in the red. We do observe the difference on those N-terminal and C-terminal peptides. In those situations, you can see in the light form, the modification level is much higher than the dark form. It indicates those regions may be involved in the interface between the two monomers of the OCP protein. And we label those in red on this OCP structures. And also, we observe some region have the reversed response, just like in the light form, it's more protected, and we labeled also in the structure. This study is still undergoing. We want to generate more structural information about this conversion between the light and the dark form, and using this protein footprinting technique, because we can directly analyze those proteins in the solution during their transition between the light and the dark form. The other technique we want to develop and answer questions is called cross-linking. This is kind of an extension of the we doing for the protein footprinting. As you may recall, in the protein footprinting, we labeled the surface solvent accessible residues by the chemical labeling. But in the protein cross-linking, we're using the bifunction reagent. That means they not only have one reactive group, they have two of them. By taking advantage of the certain length of those cross-linkers, we can form the covalent linker between those lysine residues in the complex, in the solution phase. For example, here, the cross-linker forms the two lysines. And then we use the similar bottom-up approach. That means denature the protein, digest them by the enzyme, and submit it to the LCMS proteomics experiment. Based on the sequence line we generated from the CID, the protein-induced dissociation, we can get the sequence information for both linked peptides. By that way, we can locate the interaction among the big complex and generate the networking or the multi-protein interactions between the different subunits. The other research scientist from the PARC, Hao Zhang, he was here and actually using this technique and to study the mega complex involved in the cyanobacteria. Here is the big antenna system called phycobilizome. And in the membrane, they have two reaction centers, photosystem 2 and photosystem 1. And using the cross-linking, we can generate information about how this phycobilizome actually involved and interact with the two photosystem 2 and 1 system embedded in the membrane system. We also extend this one to study the OCP protein. We're using the cross-linker to link and analyze the result to see where this OCP interact with this cyanobacteria photosynthetic system, which is the phycobilizome, the antenna system. And the result shows the OCP actually involved in the core region of this gigantic antenna system, and specifically on the APCB subunit and also the APCE subunit. This suggests the possible location of this OCP active form to interact with this antenna system to cause the cringe of the energy transfer. Overall, here I show you all those mass spec-based biophysics approaches for you to understand and further investigate those protein systems involving the photosynthetic system. That's not all we have. We actually have all kinds of different techniques still ongoing and so many targets we are working on through the PARC members and outside the PARC, and also the NIH supported mass spec center to using those biophysics tools to understand the interesting target protein complex systems. All the work I mentioned here is a teamwork. We have big teams. For example, I would thank all the former and the current members, some of them not in the picture, from the boss team, and all the money supporting us for those research, and some money from the ACL for the current development of the new technique called the nanodisk studies, and also the big team we have here for the support and discussion and all kinds of things we have here. Thank you all for the attention.
I'll be happy to answer any questions, and we also have the possibility of having questions from both sides. I don't know if you want to log on. So, who wants to be first? Mike.
I'll lead off. How could we go back to the slide that showed the discovery of the 8th pigment?
Sure.
I was interested in the slide that showed the FMO coming apart and showing what you called an unfolded. That's it.
Right.
Right. So what do you think of, it seems to me, and I'm asking for your opinion, the monomer that you formed, which has a mass to charge of around 3,500, and it has limited charging.
Right.
And also your ion mobility experiment showed that it was fairly tight. Huh?
No, it's fairly open. This one have a large distribution.
But you call it an unfolded monomer.
Right.
So, no.
If we
Well, okay, you can use that as a reference point. I'll give you the microphone, Rempel, if you join in. If we look at the ESI spectrum of the denatured spectrum, right, then using that as a reference point,
oh, I see.
so that's highly charged. Huh?
Right.
Okay, that's highly charged, and you would argue unstructured, as you say in your slide, right? Then let's go forward to the other one. The mass to charge is much higher, right? Up now 3,500. The spectrum is very simple, only 3 peaks.
Right.
And it would appear that there's no pigments in it, right?
Right. Based on the mass.
You call it unfolded,
Uh-huh.
right? So, which would seem to be consistent with the fact that there's no pigment in it.
Right.
But its mass to charge here is lower.
Yeah.
So maybe Don Rempel has an explanation for that, but I'd be curious as to what you think of that.
There's 2 reasons. The first reason is if we think about this spectrum,
Cause trouble. Go ahead, Hal. Sorry.
when we introduce this ion into the mass spectrometer, they are already denatured by the organic solvent and the acid condition. So they have the proton available to carry in this unstructured form. But the other situation, when we introduce to the gas phase, actually is the majority of the intact trimer peak, which they only have limited the proton available at the beginning. And then we apply the energy to dissociate the one subunit from the complex. So the starting point, they only have this amount of the proton available. And then we initiate the dissociation, which they take almost the half of the proton from the whole trimeric peak to be available in this monomer. That means this monomer is quite open compared to this structure. But I agree with your point is we cannot call this one fully unfolded structure. It still have some, compared to the one we have before, maybe they still have a certain conformation.
If you fast to if you go forward to the ion mobility.
Oh, yeah.
The other one. Yeah. Does this extended pattern here represent a range of conformations?
Yeah.
Where you'll have some that are more tightly folded than others.
Right.
Some are
Some
more or less
more
the same
tightly folded.
mass, but a range of different conformations, whereas the trimer seems to be rather unique in terms of just a very small range there. If you were to do the organic denatured form in the ion mobility, what would that look like? Would that also have a range?
No, they
Or
actually
would that be a tight,
also tight like this,
but it would be the monomer?
Right, but it would be the monomer and very tight because we fully denature the protein. It's the conformation actually is a very narrow distribution. But unlike here, we have a large distribution for the monomer. That means that's the average of different type of the conformation.
But you never
partial.
You never see any monomers that still have the pigments.
No, so far we didn't
Those.
see it, yeah.
So when it falls apart as a trimer, it catastrophically comes apart and loses all the pigments. At least that's what it seems to say.
Yeah, to lose the pigment. To lose the pigment. It's not highly charged.
Right. It's not at low pH. I think the pH is a big factor there.
Right. The pH in the native mass spec is 6.5.
Daryl? But I think we want people, remote people here.
Well, I noticed that your unfolded monomer section is decreasing in mass. Is it possible that on the left-hand side there is some chlorophyll with it?
I mean, here,
Yeah.
we actually didn't see any unfolding monomer with the mass shift consistent with 1 or 2 pigment attached. But here it's hard to tell from the two-dimensional plot. And that's a really good point. We should be careful and maybe do more experiment to see whether they are still attached. But we do have the situation in the other system, for example, the reaction center. When we dissociate the reaction center, we do observe the unfolded monomer with a partial attachment with 1 or 2 pigments. Yeah.
Well, it looks like you've got,
Yeah.
to my eyes, a couple of bands there that might be 1 to 2 pigments, which I was going to comment about.
Yeah, the one thing is sometimes you have those mass shift here, either because the low intensity or the signal to noise ratio on those shoulder parts, we cannot give the positive confirm about whether they are 1 or not. But we can do actually extra experiment to variable the energy to see whether we see the further shift, or we can increase the peak intensity here to confirm whether they still attach 1 or 2 of them. Yeah.
Additional questions for Hao? I have one more. If you want to go back, I can't remember which direction, to the native mass. Yeah.
Yeah.
Yeah, but go forward to the one where you had to keep going. No, where you had the mass of the pigment difference in the different forms.
Yeah.
There.
Yeah.
And one of the things that was a puzzle to us for a long time was the fact that the mass difference there is 631, which is not the mass of a bacteria chlorophyll, but rather what's called the mass of a bacteria chlorophyll-ide, which is missing the long phytal tail. And we went through a long period where we were unsure whether or not the native protein actually incorporated this modified pigment, or it incorporated the regular bacteria chlorophyll and was then damaged, or the tail fell off during the mass spec experiment. Do you want to talk about the way we resolved that? Because it was actually kind of an interesting lesson in how you have to be really careful with these systems.
Right. When we first observed this mass shift, we got confused whether we have the intact bacteria chlorophyll A here or the partial of them here. And later on, when we back to do the experiment with the isolated bacterial chlorophyll A, we realized one thing. In this structure, actually we still investigate this molecule's performance in the gas phase in the mass spectrometer. And in the regular situation, with the relatively small amount of our energy we put on the system, this tail can be easily fragmented, loose in the ESI mass spectrum. And in the native mass spec spectrum, we usually apply relative amount of energy to help us to desolvation the big complex. Usually the big complex, when we generate by the native mass spec, they can contain a couple of water molecules. To read out the accurate mass, from those complex, we need to apply the energy to help us to remove those water molecules. And those energy is more than enough to kick out this tail from the bacterial chlorophyll A. Based on those evidence, we believe that actually there's a intact bacterial chlorophyll A here. And during the mass spec, the native mass spec process. And also the one reason is this bacterial chlorophyll A is different than others inside. It's on the outside of this complex. It's very easy to receive those energy and lose this tail compared to others.
Okay. Could you go back to the slide that showed the papers that used your GEE labeling?
Yeah.
No, go forward now to the last one, to the last paper. I think Hao and Wen and Haijun and others that are members of PARC should take a great deal of pride in this paper because it was named by the Journal of Biochemistry last year. JBC, sorry, JBC, as the best paper in a certain area. And Ron Bose, who's the senior author, learned how to do this labeling from Hao and Wen and others. So I think this is a real source of pride for PARC and also for us that other people are using this and solving problems in other areas.
I might comment back on the
guess you
chlorophylls
Yeah.
paper. The section was that we've done experiments with just the bacterial chlorophyll, and we find that tail comes off quite readily.
Right.
And we've also, I verified even with theoretical calculations that that's fairly low energy process. I think I yet to publish this, but
Yeah, we're still working on that part.
Hoping
Yeah.
to get that published because I think that is a really interesting result, and it sort of is a cautionary tale in a way, in that here, in order to strip away all the solvent and so on, we have to apply this fairly energetic conditions, the CID, the collision and dissociation. And here, we're just apparently going just a little too far, at least in terms of this pigment. It just can't, it's kind of, its tail is waving in the breeze there, and it's very easy to knock it off. And so it means that you have to be careful that you're not causing some changes that, exactly.
Hao, I was just curious, now that you've developed all these really good techniques for using mass spec, and we're hopeful for the renewal of PARC, what's the next big question, or what's the next thing you would like to study?
Yeah, the first thing is not myself develop those things. We are a team. That's all the team works. We have all the colleagues here. And if by myself, I don't think I can develop all those things by myself. And the second part is you can see the mass spec is a platform. And with the different approach, like a top-down, bottom-up, you actually can further extend to all the parts. And we didn't talk about some new development we are doing right now. And with the OH radical footprinting, the nanodisk, all the new techniques. So I would like to say there are huge potential. But if we give some details about what's the next step, it's really hard to say. There's multiple projects working on it. Yeah, that's nanodisk, maybe the approach, and also the bicycles.
Yeah, doing the native mass spec on integral membrane proteins is kind of the tip of the spear, as they say, and that it's really a challenge, and we're making some progress on that with these nanodisk systems, but it's a difficult problem. Well, if there are no more questions, let's thank Hao again for a
Thank
really
you.
exciting talk.
Thank you.